It's a really great pleasure for me to introduce Professor Klaus Pantel from Hamburg. Actually, I think we don't have to introduce him into the field. Everybody knows him, but still, he is the director of the Institute of Tumor Biology at the University Hospital Eppendorf in Hamburg. He also is the chairman of the European Liquid Biopsy Society, ALPS. He is very well known in the field, a great expert since many, many years. One of his first symposia for circulating tumor cells many years ago, I was attending and I was kind of overwhelmed. I think it's at least 10 years ago. So thank you very much, Professor Pantel, for joining here into the symposium, and we're looking forward to hear you talk.
Well, thank you very much for the invitation to you, Elke, and to Ariane, and for the wonderful introduction. I think we just had finished our liquid biopsy meeting in Hamburg in May with 460 participants from 26 countries. So it's always a pleasure that you have been at these meetings, and we are going on with these meetings every 2 years somewhere else in this world. And I'm super happy that liquid biopsy has become such an interesting topic. And well, I work on it for more than 30 years, so it's a big satisfaction for me to see that, and it was not clear in the very beginning it would ever take off. So that's wonderful to see. So with these introductory words, I just show my disclosure slides. And I think Alan has already given a wonderful introduction to liquid biopsy. I can probably gain a bit of time with general slides. But I just want to mention that liquid biopsy is not only ctDNA analysis, like some people think, but it includes also circulating tumor cells and circulating other tumor cell products, such as the RNA, the microRNAs, the extracellular vesicles, and we have also included the host cells circulating in the blood, and I'll tell you why in this definition. It's clear why it can be complementary to tissue biopsy. Alan has really nicely outlined that, but I think it's also interesting to mention that the blood is really a pool of tumor cells where the products come from primary and different metastatic lesions of all locations in the patients. So it may also encompass heterogeneity in individual patients. And I think one of the most strongest arguments to me is that we, of course, take sequential blood samples every few weeks or months to really monitor the evolution of tumor and the response to therapy in a minimal invasive way. And that's, of course, complementary to what we can do with tissue biopsy. And Ellen has just showed even the single biopsy, the acceptance was below 50%. So I think we can get really more comprehensive and real-time information by the analysis of blood or other body fluids. Liquid biopsy has been also highlighted by Diana Romero in the latest Nature Milestone as key discovery, and I was very happy to see that. Where do the tumor cells and the tumor cell products like the DNA come from? Well, they're coming actually in the beginning of the disease from the primary tumor, then later on from the lymph node metastasis and advanced disease. They represent what's going on in the distant metastasis of, for example, the bone, the liver, or the brain. The blood is certainly the key analyte, and I will focus on that today, but I just would like to mention that there are also other body fluids which are interesting, like the cerebrospinal fluid or the urine in different tumor entities, and I think this will also develop in the future. In the blood, we have the cellular fraction, and in the cellular fraction, we have done a lot of work on circulating tumor cells, but also circulating immune cells and other host cells could be very interesting to add to the picture. And in the fluidic plasma or serum fraction, we have the DNA, the vesicles, the protein, the RNAs, and the tissue-educated platelets. So it's a whole comprehensive, I would say, family of liquid biopsy analytes. The two key, of course, are the ctDNA and the CTCs. And the ctDNA, I think, has been introduced very nicely by Ellen. And for the CTCs, I just want to mention that we have to do 3 steps. We can enrich the tumor cells. We can identify them by a single analysis, for example, with immunocytology. and then we can downstream analysis the CTCs at the DNA, RNA, and protein level. And we have also added models for functional analysis, including drug screening that has been published lately in EMBO Molecular Medicine. I think what is very important is also that we do a technical evaluation and technical validation of the methods. We have heard from Ellen that particularly if the variant frequency is very low, that we have to be sure that our analysis is also picking up these variants. And I think we have done such ring experiments in the context of Cancer ID, which was an EU-funded project, and then the successor consortium, the European Liquid Biopsy Society. And we have done that and are going to do that for the DNA, the microRNAs, and the CTCs in the blood. I was also part of the recommendations, I think, that were really published last year in Annals of Oncology, and Ellen has mentioned that also very nicely. And I think it's very important that these key societies publish recommendations, but it's also important that these recommendations are then really transferred into the national guidelines for treating cancer patients, because otherwise it's just another publication. We really have to work on the implementation of the guidelines into our national guidelines for treating cancer patients. In order to really bring the wonderful publications, and there are thousands of publications each year on liquid biopsy studies, but to bring these really into the benefit for cancer patients and implement it into the clinic, we have established this consortium, the ELBS. It consists of more than 70 institutions from academia and industry. We have different working groups on dissemination education, clinical studies, technology, and regulatory issues. And we bring these people together also to explain really what kind of technology is feasible for what kind of clinical question. If you're interested in this consortium, we are also open for new members. And I have a wonderful project manager, Claudia Koch, who also actually got us visited our institute last year. And please drop us a mail. Let me go to some of the applications that are relevant in the clinic. And the first one, of course, is early detection of cancer. That is very, very popular, but of course, also a very complex topic. We have initiated here at the beginning of this year a new EU project that was funded under the Cancer Mission Program. We are 17 partners from 8 countries, and I coordinate this consortium together with Matthias Löhr from the Karolinska Institute in Stockholm, and Ellen is also a member of it. So I think it's really very, very nice that we have now the opportunity to find out whether we can detect pancreatic cancer earlier, because only less than 5% of these patients have a tumor that is curable. So we need to increase this rate and make the prognosis of this aggressive disease a better one by early detecting the earliest lesions of cancer. How do we do that? We have a biobank, and this biobank is really consisting of early stage cancer patients, but also of individuals at risk to develop pancreatic cancer, which have been collected over the past 10 or more years. Then we have experts from all liquid biopsy fields, circulating DNA, exosomes, CTC host cells, proteins, and metabolites. And then we combine this information and hopefully develop a classifier that can find this devastating cancer earlier than today. Interestingly, we have also included a health economy evaluation to see how can such a test in the future implemented into the patient's management. And we have also reached out to patient organizations. This is a group here from Hamburg, and you see it's a real translational project. It involves liquid biopsy expertise of our Tumor Biology Institute. It involves clinical expertise of our colleagues at Surgery and Oncology, and the expertise of our colleagues at the Health Economics and Health Service Research. Just a few data. One set of data we have published 1 year ago. That was actually a marker, that is a circulating protein. It's a CCN1 or CYR61, which is a stress-related protein. And we could really show that it was already elevated in early stage breast cancer as compared to healthy controls, with quite a nice IUC of about 0.9. And such a protein marker could be, for example, added to a ctDNA marker. And this kind of combined analysis could increase really the accuracy of early detection. Also, I think we have just published a perspective paper, a bit provocative, and say maybe even circulating tumor cells, which have a lower concentration, of course, in early stage patient. If we increase the capture rate, for example, by increasing the volumes, we might be able to use CTCs also for early detection of more aggressive clinically relevant cancers. And I think we also have stated that CTC research can help for the understanding of tumor biology. I have no time to go in any details. So I think that also combining ctDNA analysis with CTC analysis could really give some good information. We have some years ago done a nice analysis where we looked at the molecular composition of the circulating tumor cells and the metastasis and the primary cancer and colorectal cancer to learn something about the metastatic cascade. So we got blood from the mesenteric vein, we got the liver metastasis, we got blood from peripheral vein, and of course the primary tumor. And we could really show by copy number variation analysis with each step of this metastatic cascade, there was an addition of genomic aberrations. We also have, interestingly, a great interest in really micrometastasis, so which is correlated to the field of minimal residual disease. And in this context, we have worked on the bone marrow for many years, even in the 30 years ago, together with Michael Speicher, who's also here, which is, I think, fantastic. We are still interested in the bone marrow as a source of tumor cells that can grow in the bone, but may also give rise to metastasis on the other side, so being a reservoir, and we have a DFG-sponsored consortium on that topic. The second application is risk assessment. I mean, can we use it to do a better staging of patients and decide about escalation or de-escalation of treatment? We know today that the CTC counts at the time of diagnosis are correlated to an unfavorable prognosis, so they can be actually used for studying a high-risk population and improving tumor staging. Of course, this has become also a great topic for ctDNA analysis, risk assessment, and one of the most prominent publications has been published last year by Jean-Thier colleagues in the New England Journal, and it was a question whether circulating tumor DNA analysis can guide adjuvant therapy in stage 2 colon cancer. And what they have shown very nicely is that if they treat these, if the patients had no ctDNA, these were patients with stage 2 colorectal cancer, that these patients had the same survival than the patients that received chemotherapy. And that is the big question in stage 2 colorectal cancer, which patients need chemo and which patients are overtreated by chemo. And it seems to be that the ctDNA measurements can add information to that difficult decision. The next application, of course, is some which I really like a lot, which is really following up the patients after initial therapy and prevent really metastatic relapse by early detection of the earliest signs of relapse, which really allows to intercept probably the development into full-blown metastasis. And that's shown here on that slide. With ctDNA, we can, for example, detect, of course, cancer. We can follow up patients after the initial therapy when the tumor load goes down. But we all know that there might be a residual tumor load, and that can be probably detected at very higher sensitivities than the imaging can do today. And that offers a unique opportunity for a total new field of clinical trials that we have called post-adjuvant clinical trials a few years ago with Dan Hayes when he was ASCO president. And there are really very nice studies going on. And this is a study from the group in Denmark. And I think that they have really followed up colorectal cancer patients by post-treatment monitoring of the blood of these patients. And they have shown very nicely that they can detect relapse signs much earlier than the imaging, up to 16 months earlier than imaging. And they have implemented that also now in interventional clinical trials, which I will comment a bit later. The same has been done in breast cancer. This is a study from Charles Coombs and Jackie Shaw. They've also shown if they detect ctDNA, there's a prognostic influence, as shown on the left side for the Kaplan-Meier curves. But most fascinating, they've also shown that the lead time to imaging can be up to more than 700 days, which I think gives a nice window of opportunity. And we are now performing a multi-center trial led by Wolfgang Janni called the SURVIVE trial, where we will look whether that has an impact on the mortality of cancer patients, including ctDNA in the post-operative and post-adjuvant therapy follow-up of breast cancer patients. Another, I think, focus on MRD is our second new EU project. It was funded and started in May of this year, which is a private-public partnership by the Innovative Health Initiative of the European Union. So we have academic participants and we have participants from industry. I coordinate this project together with Klaus Andersen from Aarhus University. From the industry lead, it's Maria Zaridis from BMS. We have several objectives. I think for MRD detection, we need very sensitive assay, and we have already discussed that after the first talk. If the assay is very sensitive, if we have to detect very low allelic frequency, there are a lot of things that we have, confounding factors we have to actually accept and measure. So we want to create, for example, reference standards and benchmark ctDNA assays. Then we do a robust clinical validation of ctDNA-based MRD in colorectal, pancreatic, and lung cancer as our model tumors, and integrate MRD as a potential decision tool in future treatment selections. And as you can see, we are 22 partners from academic sites and from industry, and we hope that we can tackle this very challenging question. Another application in the clinic that is very prominent is, of course, the use of liquid biopsy in the context of immunotherapies. Immunotherapies have really changed the game in non-small cell lung cancer and in melanoma and other tumor types. And we honestly focusing on this here in Hamburg. We analyze ctDNA and CTCs and actually other liquid biopsy analytes. We combine the information from these analytes. And what we have also shown lately and published a few weeks ago is that not only the tumor response is important, but also the host response. For example, we have seen that the coagulation system can affect the immune response. So we measured the von Willebrand factor, for example, in melanoma patients. So it's really an interaction of the tumor and the host that is important to predict the response to immunotherapy. And we are very happy that we were invited by Tatsuko Honyo from Kyoto to join his international network, and he's just building up this network, which is really great. And there will be also a brand new institute in Kyoto, which will be probably opened next year in the summer. So that's really a fantastic interaction that allows us to extend our liquid biopsy studies. We have just published a study in non-small cell lung cancer, and just to show that the CTCs can add some information to the ctDNA, we have looked for the expression of PD-L1 protein on the circulating tumor cells. And you can see we found this protein expressed on almost 80% of the CTC positive patients that we have analyzed here in this mixed real-world cohort. And what is really nice, and that was a study together with Martin Reck from Groß-Hansdorf. He has really a wonderful record in the lung cancer immunotherapy field. And what we could also show is intra-patient heterogeneity, which might be a very important factor for therapy escape. And as you can see here, the yellow-orange cells are the tumor cells, it's the keratin staining. The blue staining is for the PD-L1, and in the row A, you see that we have a single cell that is PD-L1 positive, but in row B, you have actually 4 cells, including a cluster that is negative for PD-L1, and the same below, a single cell that is negative, and then a cluster that is positive. So you have this heterogeneity, which probably is the greatest headache for treating solid cancer patients in general. Well, besides lung cancer, we are focusing also on melanoma, and that is our dermato-oncology group here at our last meeting in Hamburg. And you can see there can be bright blue sky in Hamburg, and in the background you also see our new opera house. So that's also an advertisement for my city. So if you come to Hamburg, don't miss the new opera house. So in melanoma, I think we have 2 cohorts that both receive immune checkpoint inhibition therapy. The one is an adjuvant cohort, that 3 patients which are then monitored very densely. So we get blood samples every few weeks, and we have a metastatic cohort that is also monitored densely over time. And we analyze ctDNA, but also other liquid biopsy components. We have established a multi-center biobank, and that is really thanks to the wonderful clinical scientists shown on the last pictures that really do a lot of work to establish this biobank. And this is really a great asset for our ongoing and future collaborations. We have, for example, started some years ago to also assess the ctDNA method from Argentina, which is not based on any bioinformatic or engineered information. And we have shown that with that, we can get a decent sensitivity, and the results come quite quickly. Of course, it's only a limited panel, in this case, 13 genes that are melanoma relevant. More lately, we have some unpublished work, also working together with the assay here from Sysmex. This is a melanoma plasma assay that looks at 5 different genes: BRAF, EGFR, KRAS, NRAS, PIK3 kinase. We have then really used this assay in our dense correlation of metastatic melanoma patients and compared that to the usual parameter relevant for monitoring metastatic melanoma patients. And I have no time to go in big detail, but just to show if we really find DNA, patients have a worse outcome, which is shown here at the Kaplan-Meier curve. We just a few examples. We can also see that we detect the rise in ctDNA earlier than we detect the progression with the radiology analysis. On average, in this metastatic cohort, 4 to 12 weeks before. And of course, we have then moved on to a larger panel in collaboration with Personalis, where we have done now a tumor-informed assay, where we analyze up to 1,800 variants. Then we do a tumor variant tracking over time. And we have also started to do that in our melanoma cohort and go on now with our adjuvant cohort in the future. And here you see that we can get a whole range of genes. These are not only melanoma genes, of course, but all genes that we have really being tracked. And we have also seen that if we have a ctDNA clearance, that these patients do better than the ones that have no clearance. But again, that's just a pilot study on 25 patients, many samples, but just a few patients. So we have to, of course, validate that. Well, in order to bring really liquid biopsy into clinical practice, we need to do interventional clinical studies to get clinical utility. And I think this means that we have to change the patient's management based on our liquid biopsy analysis. And then we have to prove that this change is really doing something good for the patients in terms of survival, life quality, or any other benefit. And these studies are going on, and this is an example for colorectal cancer from my friend and colleague Klaus Andersen and his colleagues in Aarhus in Denmark, who's coordinating a nationwide study on that, where they monitor the blood after the initial treatment. And for those patients that are positive, I think they look where they can find actually the metastasis, and those that are negative, they go on with the serial ctDNA analysis. And of course, the big question is, whether these patients profit from this kind of monitoring in terms of survival, overall quality of life, and they also look at cost effectiveness. We have done a study on CTCs, which also was interventional. It was already mentioned, the HER2 oncogene has become again a great target for therapy. Even the HER2 low patients are now interesting. So what we have done, actually, we have typed CTCs for HER2 expression many years ago, and we could see that there's a switch of the HER2 study from the primary tumor to the metastatic relapse time, which there may be many years in breast cancer between primary tumor treatment and metastatic relapse. So in some patients, this HER2 status can change, and we can analyze that by assessing the CTC HER2 immunocytochemistry status. So we have then really decided years ago to do a randomized interventional trial and treat the patients that are positive for HER2 on the CTCs at the time of metastatic relapse, but were negative at the initial primary tumor time. That these patients received an anti-HER2 therapy, and lapatinib was chosen for several reasons. And this is the unpublished result of the overall survival analysis. So you see the red curve are the patients that received the standard therapy plus lapatinib, and the blue curve, the patients that received only the standard therapy, and there was a significant improvement. So I think that is the first randomized interventional clinical trials indicating or suggesting some utility of characterizing CTCs. In the future, I believe that we will see new liquid biopsy components, and particularly the extracellular vesicle field is really evolving very rapidly with a beautiful international society that also cooperates with our ALPS. And EVs, of course, are also functional LB marker. They are released actively from tumor cells and host cells. They are taken up by recipient cells, and they can change the biology of the recipient cells. And we have shown, for example, together with David Leiden years ago, that the integrin content of exosomes can guide organ-specific metastasis by building really organ-specific niches for metastatic cells. So this is, I think, an interesting new component. And we have, for example, in lung cancer patients analyzed 1.5 ml of blood. And we can now, together with our mass spec facility, analyze more than 5,000 proteins from this small volume of plasma. And then we can do, of course, a functional annotation. And it really shows how much potential is also in this field. However, this is also a field where harmonization of technologies is extremely important to actually obtain reproducible results and make any implementation in the future in clinical decision making. But I think that the groups are on their way to do that. And we have been participating in a Marie Curie network, which was also coordinated by Tom Würdinger of Amsterdam. So in conclusion, I think early detection of primary cancer NMID relapse require very sensitive assays. The assays need to be validated and harmonized. The assays can provide clinically relevant information, but we have to get, of course, sufficient data and know the confounding factors. We believe that the combination of different LB biomarkers may lead to better accuracy, and the resistance to immunotherapy may depend on both tumor and the host response. And of course, last but not least, we need more interventional studies to demonstrate clinical utility. These studies are ongoing, and some of them have been finished, which is really great. And we are talking today with the EU-RTC resist groups, which is actually very good, because this interaction may lead eventually to the implementation of ctDNA into the RECIST 2.0 criteria. And that is an ongoing project, which I think has great potential for future use of liquid biopsy in clinical decision-making. So at the end, a big thank you to our network here in Hamburg, which consists of research institutes and clinics that treat cancer patients. An even bigger thank you also to the people at the Institute of Tumor Biology. We are 50 to 60 colleagues from more than 15 countries, a bit like United Nations. And a big thank you to the funding organizations that fund 80 to 90% of our research, and particularly the ERC, but also the other European organizations, which have been of great help for us, and also the German organizations that support our research for several decades now. And that's it. And thank you very much for your attention. And I'm now open to answer your questions.
Okay. Thank you very much for this comprehensive overview. Wow, what a force ride through all your projects and results. There are no really important questions. Now one I will tackle in my questions. We have many clinicians and here in the audience and people that are not heavily involved into this topic. And so let me ask just one really simple question or summarize something. So liquid biopsy touches cell-free DNA, of course. This is the major topic today, but it also touches circulating tumor cells and exosomes. And exosomes are kind of part of the tumor cell, but they can't live on their own. And circulating tumor cells, as I have learned, they kind of travel in schools. So they're not alone. They travel in together with other tumor cells and other normal cells, actually. How frequent are these little tumor packages in a blood sample, let's say, of a patient with colorectal cancer stage 2? So that people get an impression of what's going on if you have already going on in your blood, if you have an early stage cancer.
Well, I think that most of the tumor cells travel alone. They are also packages or clusters, which are maybe 10% or less of all the positive patients. So I think that most cells travel alone. And in early stage colorectal cancer, like stage 3, you have around 20% of the patients that have these cells be detected. If you have a normal vial of blood, if you take more blood, you can increase the frequency. And many people have shown that that is a prognostic factor. So you can really use it, for example, in stage 2 tumors. We have published that 10 years ago, that in stage 2 tumors, you can have two stage 2 groups, one with a bad prognosis, one with a good prognosis. Very similar to the New England data that were published 10 years ago with the ctDNA. So I think in that case, yes, you cannot use it for early detection of cancer yet, but of course for identifying high-risk groups, like stage 2 probably, that could or could not need more therapy.
Yeah, exactly. Stage 2, which is not truly stage 2.
Exactly.
How you get them out of the blood sample?
Well, I think we have different methods. In principle, we can get them out with antibodies connected to cell surface antigens, such as EpCAM, but we also can get them out based on size selection, and that's also two very complementary methods. And some tumor cells do not express their surface antigen that you take out. Some tumor cells might be too small for the size selection. So I think both methods are really complementary.
That was one question from the chat.
I saw Christoph has a question.
No, that's the hand a leftover hand from the talk before.
Great.
But we are now perfectly in time. I thank you very much for this overview, and we've learned a lot, and we see what's coming on in the near future from all these very many huge projects. So there will be benefit for the patients in the future, I'm sure.